So during the first century of neuroscience, there were many attempts to control the brain. For example, you can put an electrode into the brain and stimulate with electricity and get pretty well-timed responses. There's a problem though, which is that electricity goes in all directions. It's hard to know when you electrically stimulate the brain exactly which parts of the brain have been recruited. So what we decided to do was to develop strategies to allow light, which can be aimed at cells, to stimulate neurons. And of course, neurons don't normally respond to light. So for me, what really triggered the idea of going after this was a paper that I found, which was published by a group from Japan in 1999. And they were surveying these light-driven proteins that occur in archaea, single-celled prokaryotic organisms. And normally they're found in very halophilic archaea, that is, prokaryotes that live in very, very high salt concentration environments, like the Dead Sea or the Great Salt Lake or other very high saline environments. And so, of course, if you took those molecules and put them into a neuron, they wouldn't work. But this group found this one organism actually has molecules that work in low salt concentrations, much like those that you would find in your brain or in my brain. And so if you take those molecules and put them into neurons, what we found is that you can actually use them to control the neural activity. So in optogenetics, we put a genetically encoded molecule into a neuron that converts light into some kind of electrical signal, and then we shine light on that neuron. So light gets converted to electricity, electricity controls the neuron, and now we can control the neural activity with light. Many groups have been using optogenetics. For example, one group has found a small cluster of cells deep in the mouse brain that when you optically activate them causes aggression. A mouse will attack something next to it, even just a regular old glove. Another group has been studying smell, and they've been able to show that if you activate cells in the olfactory cortex of the brain, that you can associate those olfactory synthetic stimuli with positive or negative outcomes, rewards or punishments. So smell might be a very plastic sensory modality. And a third group has been working on ways to try to treat blindness. One of these forms of blindness, retinitis pigmentosa, is caused by over 100 different genetic mutations. So if you try to fix all those mutations one by one, it might take a long time. If, on the other hand, you can insert light-activated proteins into the spared cells of the eye, converting them into a camera essentially, then you might be able to cure many patients all at once. So several groups have now been experimenting with mouse models of blindness, showing they can restore function. Optogenetics hasn't been used in any clinical scenarios to date. There are many people interested in human clinical trials in the near future, but so far all of the studies have been done in animal models, preclinical work, as it were. Well, if you really want to solve the brain, we have to do more than just control the brain. We have to be able to record the brain, map the brain with anatomical and molecular resolution, and ideally be able to build brains from scratch. And so, often working in multidisciplinary collaborations, we're working on tools that let us do each of those things. I would love to see maybe sometime in the next 20 years, optimistically, 40 years pessimistically, maybe around 30 years, that we can completely understand the brain. We understand how it computes thoughts and feelings, consciousness and awareness. And ideally, we would have such good maps of the brain that we also can achieve good handles, or at least know the directions that we need to go into in order to cure all brain disorders. So that's the mission that motivates our group, is to build the tools that let that happen.